I would like to start by thanking the IMP and the Birnstiel Foundation for honoring me with this amazing award and the opportunity to present my research. I'm sincerely grateful for that. During my PhD, I studied the role of a mutant protein called p53 in the development of colorectal tumors and found an interesting linkage between colorectal cancer, mutant p53, and our gut microbiome. p53 is a highly conserved tumor suppressor gene which responds to cellular cues and protects the cell from malignant transitions. And the way it does it is by promoting the expression of various target genes that stop cellular division by inducing cell cycle arrest, senescence, or apoptosis. Due to its efficient tumor suppressive function, p53 activity is disrupted in the vast majority of human tumors, usually by loss of function mutation that prevents its ability to interact with the DNA and express its target genes. The loss of function mutations are often endowed with gain-of-function properties that enhance tumorigenesis even beyond the loss of wild-type p53 activity. Colorectal cancer is one of the most common malignancies in men and women. In most cases, colorectal tumors arise with a mutation in genes related to the mitogenic pathway called the Wnt signaling pathway, resulting in overactivation of the Wnt pathway, accelerated cell proliferation, and the growth of small benign adenomas. Mutations in p53 are usually seen later in the sequence of mutations, turning the polyps into invasive carcinomas. Interestingly, even though the small intestine is 2 to 3 times longer than the colon, small intestinal malignancies are very infrequent, and the reason for this rarity is still not fully understood today. To decipher this enigma, we generated an intestinal cancer mouse model in which a protein called casein kinase 1 alpha, or CK1 alpha in short, is knocked out specifically in the gut epithelium. CK1 alpha is a negative regulator of both the Wnt pathway and p53. Thus, its deletion results in overactivation of Wnt in the gut and stabilization of p53, which maintain a fragile gut homeostasis. When both CK1 alpha and p53 are coablated, mice suffer from high-grade dysplasia and succumb to death eventually. By breeding the CK1 alpha knockout mice with mice harboring one of the most common gain-of-function mutations of p53, the R172H missense mutation, we were able to examine the gut pathogenesis when Wnt is high and p53 is mutated, similarly to human colorectal tumors. CK1 alpha mice that harbor either wild-type p53 or gut-deleted p53 were used as control groups. Here is an immunostaining for the proliferation marker Ki67 in wild-type mice. This is the staining in the jejunum, which is the proximal part of the small intestine, and in the colon. In the small bowel, the proliferation is located mainly in the stem cells, which are at the lower compartment called the crypts, and these are the differentiated villi. In the colon, there are no crypts and villi, but a bottom layer of dividing cells and an outer layer of differentiated cells. In CK1 alpha knockout mice that carried the intact p53 gene, homeostasis was maintained due to the stabilization of wild-type p53 and its tumor suppressive activity. On the other hand, co-deletion of CK1 alpha and p53 resulted in high-grade dysplasia and tumorigenesis in the whole intestinal tract. Interestingly, mutation p53 led to an opposing phenotype at different gut segments. While the colon, as expected, was highly proliferative and dysplastic, the small intestine showed normal proliferation with no evidence for high-grade dysplasia, suggesting a novel tumor suppressive activity of p53 solely in the small intestine. To unravel this tumor suppressive activity, we checked the two pathways affected by CK1 alpha deletion, Wnt and p53. Examination of the p53 pathway revealed that although it was highly stabilized upon CK1 alpha deletion, mutant p53 loses its transcriptional activity and similarly to CK1 alpha p53 double knockout mice, no p53 target genes were expressed in the mutant p53 cohort. To study the effect of p53 mutation on the Wnt pathway, we checked the expression of several Wnt target genes, and as expected, mild activation was seen in CK1 alpha knockout mice, while in CK1 alpha p53 double knockout mice, a robust activation was detected. On the contrary, mutant p53 mice showed a clear reduction in Wnt activity, even beyond the reduction seen in CK1 alpha knockout wild-type p53 mice, with expression levels similar to wild-type mice. By contrast, Wnt activity was very high in the colon of mutant p53 mice, suggesting a differential activity of mutant p53 at different gut regions, tumor suppressive in the small intestine and oncogenic in the colon. Our body is a complex ecosystem with an enormous number of various microorganisms. The gut is the most heavily microflora populated organ in our body, with an increasing microflora gradient from the small intestine to the colon. Since mutant p53 inhibits Wnt solely in the small intestine and not in the colon, we hypothesized that gut microbiota may handle Wnt suppression by mutant p53, specifically in the colon. To test this hypothesis, we eliminated the gut microflora of our mice by treating them with a cocktail of antibiotics and compared the colon to vehicle-treated animals. Notably, the treatment affected only the mutant p53 mice, which showed the suppression of Wnt and therefore reduction in the colonic dysplasia rate and proliferation capacity. Altogether, these data suggest that gut microbiota may oppose Wnt suppression by mutant p53 and therefore promote tumorigenesis in the colon. One way by which gut bacteria affect the host cells is via secretion of metabolites. These food byproducts are observed into the cells where they can modify the activity of proteins in the host. And in an ex vivo setting of gut organoids, in which we tested the effect of different bacterial metabolites, we found that gallic acid, which is a polyphenol-derived metabolite, highly enriched in black tea and coffee, increased Wnt activity and proliferation capacity. Notably, the effect was seen only in CK1 alpha knockout mutant p53 organoids and not in other lines. Real-time PCR analysis confirmed that gallic acid-producing bacterial enzymes were indeed present in the intestine of our mice with significantly higher levels in the colon. To check the effect of gallic acid in our in vivo setting, we treated our mice with gallic acid and compared their small intestine to non-treated mice. Interestingly, gallic acid treatment affected specifically the small intestine of mutant p53 mice, which became highly proliferative and dysplastic with high Wnt activity. Finally, we co-treated our mice with antibiotics and gallic acid and found that gallic acid completely abolished the antibiotics effect seen in the colon of mutant p53 mice, indicating that gallic acid can fully reproduce the effect of gut microbiome in abolishing the tumor suppressive activity of mutant p53. Altogether, our data suggest that the gut microbiome may suppress the tumor suppressive activity of mutant p53 in the colon, and we demonstrate a novel tumor suppressive activity of mutant p53 in the small intestine, which may give a possible explanation for the rarity of small intestinal tumors. With that, I would like to finish and thank my supervisor, Professor Inoon Ben-Aria, all my lab members and collaborators, and thank you very much for your attention.